All right, so enzymes. So first off, if it ends in ASE, it is definitely an enzyme, okay? Not all enzymes end in ASE, which is a little bit confusing, but if it ends in ACE, then it's an enzyme. And please be careful of your spelling. So on your exams, if you spell it and it looks like lactose, but the question asks you to name an enzyme, you'll get it wrong because lactose is a sugar, but lactase is a protein enzyme that breaks down lactose. Usually the prefix on the enzyme tells you the substrate. So lactase, an enzyme that breaks down lactose. There's also amylase, an enzyme that breaks down amylose. Lipase breaks down lipids. Nuclease breaks down nucleic acids, and there's a ton of them. But you also need to know the definition of an enzyme and be able to like spit that out on a test. So an enzyme is a biological catalyst. You should have gotten catalyst last year in chemistry class, but a catalyst is some molecule or ion or atom that speeds up a chemical reaction without being affected by that reaction. So catalysts or enzymes can be reused. And what they do is they speed up the rate of the chemical reaction so that life can happen, life can exist. Like if we didn't have enzymes, we probably wouldn't be around to be talking about enzymes. Okay. Proteins, so structurally, an enzyme is typically a protein or an RNA molecule. We're actually finding out more and more that RNA acts as an enzyme, but that's not in our curriculum yet because it's still kind of new research and it takes a while to get put into the curriculum. So that's the structure. So like what macromolecule class it falls into, but enzyme is the function. You do need to be able to recognize this graph. So this, you'll notice there's two lines and the starting point and the ending point are the same. Okay, what this is representing down here is time, and then on the y-axis is energy. Okay, so the starting and the ending point of the reaction is the same with regards to the energy, but what happens is the enzyme reduces this curve. So this is called activation energy, which I don't see written here. I'll write it up here so you can see it. Activation energy. All reactions need activation energy to happen, whether it's an endo or an exothermic reaction, okay? They all have to have activation energy. You can think about it like a person who wants to start a small business. You have to have capital, you have to have money to start your small business. Well, if you need, I don't know, $50,000 and you're going to save it up yourself, it's going to take you a really long time to save up $50,000 to start your small business compared to if you went to a bank and got a loan. So that in this case, the bank would be like the enzyme. If you get a loan of money, you reduce activation energy needed. So you don't have to physically wait to get your money to start your business. So you could be given this picture on the exam and you might have to, I've seen it where they just give you this curve without an enzyme and they ask you to draw it in with the enzyme present. You just draw a line that's similar in shape, but the curve is lower. The start and the end have to be the same. Or they might draw, these different arrows in and ask you what's going on here. So you do need to know and use the vocabulary. That's the same throughout all biology. It's like learning another language, literally. Like there have been studies on biology where you learn just as many, if not more, vocabulary words as you do when you're learning, say, Spanish or French. You need to know enzyme and you need to know substrate. So in a chemical reaction, you start with reactants and you end up with products. So when we're talking about enzymes, the reactants are the substrate. You have a chemical equation, the enzyme is not actually represented in the equation because the enzyme doesn't change. What will happen is the substrate will bind to the enzyme's active site. That's another one you need to know, active site. Has a complementary shape, both physically and chemically. They're not bonded, it's not a covalent bond, it's binding. So there's like a polar attraction. With the substrate bound to the enzyme, we call that the enzyme substrate complex. And the enzyme can reduce the activation energy by stretching and pulling on the chemical bonds here, if it's a larger substrate that's being broken down, or by placing two substrates in closer proximity in the right orientation, it reduces the energy needed for their reaction to take place. Once the reaction takes place, you have the product, it's a different chemical now, so it's ejected from the active site and the enzyme can be reused. Now, enzymes are substrate specific, but there's a little bit of wiggle room. So we call this the induced fit model. That's kind of like how more than one person can wear the same pair of gloves. Gloves kind of stretch out a little bit to fit the fingers that go in them. So the active site can chemically stretch or change its shape a little bit to accommodate the substrate. And there's some enzymes that can catalyze more than one reaction, but it's a narrow set, and those substrates are similar in chemical shape. Next slide. So these are examples of enzymes. You are required to give examples of enzymes. You don't have to know topoisomerase by name, but it is an example. You do have to know Rubisco by name, and we'll be talking about specific enzymes as we go along, especially when we get to digestion, cell respiration, and photosynthesis. So topoisomerase is a named example. This is a picture of it. This is kind of zoomed out using a computer imaging. This is a ribbon model for the protein. You can see there's a hollow space inside, and that's the active site. Topoisomerase is involved with untwisting the DNA double helix, and it works along with helicase. So it does make a temporary cut in the helix to start untwisting it, and then it helps to like stabilize the helicase because there's a lot of torque going on during DNA replication. Okay. Then you see this one is Rubisco, and we will come back to this one by name in this chapter when we talk about photosynthesis. Rubisco is an enzyme that's involved in the Calvin cycle of photosynthesis. So what it does is it attaches carbon dioxide to some precursor molecules that will eventually make sugars from photosynthesis, like glucose. So when you look at the organelle that is the chloroplast, if you were to dehydrate it and then take the mass, half of that mass will be Rubisco. Okay, it's really important to photosynthesis. It's highly inefficient. It's one of those evolutionary quirks. Sometimes it doesn't do exactly what it's supposed to do, and you get a toxic byproduct of carbon monoxide instead of the carbon dioxide. But it works good enough, and it was good enough compared to all the other enzymes around at the time when the chloroplast first evolved, so it stuck around for us. Okay, now this is what I had a question on earlier. So enzymes, in addition to having inhibitors that slow them down or turn them off, they also have other molecules called cofactors or coenzymes that turn them on. And these, along with inhibitors, act as control mechanisms to make sure the enzymes are only functioning when needed, so that the cell or the organism the cell is a part of isn't wasting energy or time or materials doing some chemical reaction that is not needed. Like if you're not sick, you don't need to be making antibodies, for example. So I'm going to write this in since it's not on the notes. There's also coenzymes. So the difference between the two, a cofactor is a mineral, and then a vitamin is a coenzyme. So like from a chemical standpoint, coenzymes are organic. So the coenzyme is carbon-based, whereas the cofactor is inorganic. It's going to be an ion of some sort. So for example, in your blood, we have hemoglobin. Sorry, that's not a proper example. Well, there's one here. Magnesium is the cofactor for rubisco. So without magnesium, plants cannot grow. So if you actually look at fertilizers, that is one ingredient of fertilizers. And if you ever look at a multivitamin, it also contains minerals in it. And we humans do need trace amounts of iodine. We need trace amounts of copper, zinc, things like that to act as cofactors. Coenzymes, these are going to be our organic vitamins. So vitamin A, all the many vitamin B's, vitamin C, vitamin D, vitamin E, and vitamin K. They weren't named in a logical fashion, but they all act as coenzymes. So a coenzyme would be vitamin C, like I told you about earlier. We need vitamin C to catalyze the reaction that makes collagen. And vitamin C acts as a coenzyme in other reactions as well. So cofactor, coenzyme, they act to turn the enzyme on. Okay, we'll come back to this too when we talk about mitosis and cancer, the cell cycle. We have coenzymes that interact with enzymes that control cell growth. So we actually have, well, we'll get to that later. I don't want to confuse you now, but you have a coenzyme that interacts with a protein that tells your cells to go through cell division. But if you have cancer, that changes that balance. So your cells just constantly go through cell division instead of stopping as they're supposed to. Okay. So evolution does act on enzymes because the enzyme is either a protein or an RNA, and those are made from DNA, a gene. So evolution does play a role in enzyme structure and function. You can have a family of enzymes that are a little bit different. We actually track these relationships like across like different species of, say, animals to check on like relatedness patterns and see how enzymes have mutated over time. There have been lots and lots of studies, but we can compare the genes for the enzymes, or we can compare the amino acid structure of the enzyme if it's a protein enzyme to check on like how divergent a species is from a sister clade. So when you see in the news, if you hear that, oh, we found out this dinosaur gene or something is different from these other dinosaurs, they're not actually looking at dinosaur DNA because DNA does not last that long. So there's one reason why Jurassic Park wouldn't quite work. But we have found dinosaur proteins. a lot of collagen because that's in bones, but you can analyze proteins in a similar fashion to DNA and do genetic comparisons with the proteins. Okay. Later on, when we get to evolution and genomics, we will be looking at hemoglobin. It's not an enzyme, but the hemoglobin will, it's studied a lot because it's found in many species and it's a pretty tightly conserved molecule. So they look at the differences between it. Okay, so enzyme regulation. So one way to regulate the enzyme is to have an enzyme that's always off unless it's in the presence of a cofactor or coenzyme. Another way to regulate an enzyme is to have an enzyme that's always on unless it's in the presence of an inhibitor. Okay, so you can have competitive or non-competitive inhibitors. Thankfully, these are named appropriately. These make sense. So on the enzyme, you have, move this up a little bit, you have a spot, the active site where the substrate goes. But many enzymes also have another spot that's called allosteric site. S-I-T-E, okay? So a competitive inhibitor will actually block the active site. So the substrate cannot bind and the enzyme is not catalyzed. So this is the example I gave you earlier of a defender, smacking a basketball away from the basketball hoop. Okay. There's still some reaction taking place because there's a lot of enzymes, a lot of substrates, a lot of inhibitors. So just by random, these all kind of bounce around. So you do get some reaction, but it's really, really slow. But a non-competitive inhibitor is an inhibitor that binds to the allosteric site, and it actually causes what we call a conformational change in the enzyme. Not conformation, conformation, which is a shape change. You can have a conformational change in any protein. In fact, most proteins have more than one conformation. So the non-competitive inhibitor causes a conformational change of the active site, so the substrate cannot bind. So that was, like I said, with the basketball, if someone like sewed together the basketball hoop so you couldn't, or the basketball net, so you couldn't make a basket and score a point. That's the difference. The non-competitive inhibition is reversible. When the inhibitor goes away, the active site will bounce back to its original shape. But as long as it's there, this enzyme is not catalyzing any reactions. Okay. Cells actually use this as a method of control. And a lot of enzymes work in a cycle or in a chain. So you have one enzyme that the products of its reaction is the substrate for a second reaction and a second enzyme, and then the product of that is the substrate for a third enzyme reaction. So a lot of times in these metabolic pathways or metabolic cycles, the end product will inhibit the first enzyme. So, if we're making, say, ATP breaks down if you don't use it. So you can't just stockpile tons and tons of ATP. It'll go bad, kind of like you can't just stockpile a bunch of tomatoes. They'll rot eventually. So ATP will act as an inhibitor for the first enzyme in the pathway to make ATP. So when you have a lot of ATP, you stop making ATP. So this is showing you, again, the allosteric other site. So it can work. Allosteric interactions can work with a coenzyme or a cofactor, or it can work with an inhibitor. It just depends on the specific enzyme in the case that it's in. So some enzymes need a cofactor to turn on. Some need an inhibitor to turn off. This is just extra. A lot of proteins are made of separate polypeptide strands, and we call those subunits. So enzymes that are proteins can also have subunits. So there are some cases where one coenzyme or cofactor can turn all of the polypeptide subunits on or off, depending. Okay, so here's another example of an enzyme. It's called caspase. This is the enzyme that's responsible for cell death. So sometimes cells need to die for the greater good of the multicellular organism. So there's apoptosis is when a cell is told to basically commit suicide, and it's done with caspase. So the cell will start basically digesting itself, and it'll contract its plasma membrane in and form these little things called blebs, and it'll pinch off, and eventually it will die, and then other cells in the organism can recycle it. So that happens normally with our immune system. It will identify an infected cell, or if there's a cancerous cell, it'll use caspase to trigger cell death. Apoptosis also happens during embryonic and fetal development. So for example, we humans, when we have our limb buds start forming as a fetus, our hands actually look like paddles. And their enzymes, the caspase triggers the cells that would be a web between your fingers to die off so that your fingers, can move. So the webbing grows, and then the caspase triggers apoptosis there. Sometimes it doesn't work for genetic reasons or birth defect reasons, and you have people born with webbed hands or webbed fingers. But there are, this is in the textbook that we're not using, but this is a further case study of the experiments they did to figure this out. Now, necrosis is cell death where the cell dies from an outside agent killing it, like if you get infected by bacteria, if the cell gets infected by bacteria and it kills it. All right. So eukaryotic cells are compartmentalized. They have organelles. Those organelles are membrane bound except for the ribosomes. They're membrane bound so they can control their own optimal conditions on the inside. So we don't just have, in a eukaryotic cell, you don't just have enzymes running around willy-nilly digesting the cell itself. Okay, everything's tightly regulated and controlled. So inside the mitochondria has its own special pH and its own enzymes and its own substrates. We'll talk about the anatomy of the mitochondria in a little while when we talk more about cell respiration. Lysosomes too, they contain digestive enzymes that are really important to keep the cell safe from any bacteria that happen to infect it, for example, but you don't want the lysosomes digesting organelles that it needs to survive. Enzymes aren't sentient. Okay, these are just chemicals floating around, and anytime a lysozyme sees its substrate, it'll do the reaction. So it's kept in that organelle to compartmentalize it. Okay, now we looked at these graphs already on your POGIL activity, but environment does affect enzyme function. So enzymes have evolved to have an optimal pH and an optimal temperature. So for multicellular organisms, typically the optimal temperature is going to be the same for every cell in that organism. So for humans, our optimal temperature is 37 C. That's body temperature. You can see that here. If the temperature is lower than that, the enzymes can still function, but they'll function at a slower rate. If the temperature raises above that, the enzyme will denature and it's no longer functional. So if it gets too hot, it's irreversible. You can't go backwards. Okay. But if it's too cold, the enzyme's just going slow. It's not denatured, so it's okay. But each different organism has its own different optimal temperature. So this is for a thermophilic bacteria, heat tolerant, so its optimal is at 77. Okay. Now some animals can't regulate their own body temperature very well, so that's why you see, for example, alligators lying on the side of the road or by a lake or something when it's cold. They're using the sun's heat to bring up their body temperature. And I mean, don't do this, but they're actually quite lethargic when it's cold because all of their enzymes are operating at suboptimal rates. So it'd be kind of like they're groggy. They can't move or do much. So that's why they lay out in the heat. Snakes too. That's why a lot of times you see run over snakes on the road because the asphalt is warmer than the ground. So they're trying to warm up. Don't go messing with alligators though. Now, pH is different. pH, anytime you deviate from the optimal pH, the enzyme will denature. So if it's too low or too high, the enzyme will denature because pH is a chemical change and you have different ions interacting with those bonds that hold the protein enzyme into its quaternary structure. So it'll snap, the hydrogen bonds, the disulfide bridges, the ionic bonds, and the interactions. An example of an enzyme denaturing with heat, you can think about cooking. When we cook eggs, for example, there are proteins as well as enzymes within the egg white. So the egg white, as you add heat and you pull it above its optimum, you'll denature the proteins in the egg white, and the egg will go from clear to white, and it's irreversible. You can't put the egg back. Okay. You could do the same if you had a strong pH acid. If you crack an egg and put it in, say, a hydrochloric acid, the egg will also turn white. I would not recommend eating it. It would be bad for you, but it has the same effect. Now, within the organism, there are different pHs for different enzymes. So our stomach is highly acidic to hopefully denature any pathogens we eat and also to help us start digesting our proteins in our meal. But then in our intestines, that pH is around about 8. And in our mouth, it's about a little bit higher than 7, but not much. So we have different pHs as you go through the organism's body. And then within the cell itself, different organelles have slightly different pHs as well. So these are enzyme specific. So if it's an enzyme in the stomach, it will always be 2. We'll come back to this when we talk about nutrition and digestion. In the intestines, it's always going to be 8 as the optimum. And here's the graphs again, okay? So notice that the pH curve is a little bit more like a bell curve, but the temperature curve, you get a pretty much a line until you reach optimum and then a sharp decline. So you think about this one more like a, I don't know, like a mountain slope if you, and then this one down here, if you're asked to describe this, well, at low substrate concentrations, as you increase substrate, you increase enzyme activity rate up to a point where every enzyme becomes saturated, and then the reaction rate plateaus. The reaction is still going on, but the speed of reaction is not getting any faster. You will be asked to identify these graphs or to, like, you'll be given pictures and asked to label which is which, or they'll tell you, oh, like draw a pH for an enzyme that has an optimum pH of 8 or 6 or whatever it happens to be. Okay. And this is what I was talking about earlier. There is something called feedback. So most of the reactions in our bodies are actually what we call a metabolic pathway or a metabolic cycle or a loop. So you have this enzyme, in this example, the substrate is threonine. Threonine gets catalyzed by this enzyme that turns this substrate into a different product that gets catalyzed by enzyme 2, and then the product of that gets catalyzed by enzyme 3, and that product by enzyme 4, and that product by enzyme 5. So by the end of this pathway, you get isoleucine. These are different amino acids, by the way. And the isoleucine acts as a non-competitive inhibitor for this very first enzyme. So it turns it off. So when you have a lot of isoleucine, you don't need to make more, so it's turned off. This is called end product inhibition. End product down here, and it's inhibition, or sometimes it's feedback inhibition. Some of our medicines work in this fashion. So we have medicines called ACE inhibitors. ACE inhibitors. ACE, which are non-competitive inhibitors. And what they do is they function to lower blood pressure. So people who have chronically high blood pressure, doctors will prescribe them an ACE inhibitor, and it inhibits the first enzyme in a pathway that keeps your blood pressure high. They're fabulous. Great. It's been a boon to people to help them survive. So those are, and this feedback, it's all non-competitive. Okay. Non-competitive. So in this case, when there's no more isoleucine, the pathway gets turned back on and more isoleucine can be made. In this case, if you stop taking your ACE inhibitor, your blood pressure will go back up. So it is important, if the doctor does prescribe you medications, that you stay on it and consult with your doctor. Don't just quit it, because you'll bounce right back. Okay, we're all finished with the notes.